Hello and welcome to BioCode. As seen in previous video, you've seen that we introduced alphabet class from the BioPython. In this video, we will see how we can utilize various functionality that is provided by the BioAlphabet to understand the DNA or protein, generic alphabets or generic DNAs. Basically, we need to define basically alphabet. A class of BioPython packages utilized to define our sequences, define our strings, define our whatever sequences that we are providing to the Python language. So to actually define them, we need to import those functions from the BioPython language through the alphabet class. In the previous video, we also imported the alphabet class module, but in this video, we will be importing IUPAC, and that is also a module within BioAlphabet that allows us to demonstrate or define those sequences that should be defined as ambiguous, unambiguous, and that sort of scenario. So another cool thing about BioAlphabet is that we can also directly import DNA alphabet, protein alphabet, generic alphabet, generic DNA, generic protein. I will describe each of them, and you need to understand all of them. Generic protein basically let's come from the top of the chain. DNA alphabet basically means that it is a DNA alphabet, and all of the sequence that we see, that basically it is you telling the Python language that this sequence is DNA, and all of these sequences DNA alphabet, whatever it contains, it should be a DNA, right? After that, we have a protein alphabet, and you are telling the same thing to the Python language. In this case, it is going to be a protein. Generic alphabet can it mean it is you telling the Python language that it is anything. It could be a protein, it could be a DNA, it could be anything. So it should not consider it as, it should not put a restriction or strict restriction on it that it is a DNA, it is a protein. It can be anything. It can contain DNA sequence, it can contain a protein sequence as well, and it can contain merged sequence as well. For example, let's suppose the 50 amino acids plus 50 nucleotide sequence, which is not basically utilized in professional life of bioinformatics, but just to provide you an example, it is very useful when we are working with various various sequence files, and we don't know if some of them are protein files or not. Okay, so generic DNA also defines that it could be a DNA, but it can have some issues with it. For example, there could be NNN, there could be some those sort of nucleotide representation that are not in the DNA alphabet. DNA alphabet will always be ATGC. Generic DNA can contain other sort of representations of the DNA. You can read more about this in the IUPAC article of ours. Generic protein, basically, as I said earlier, it means the same as DNA, but in this case, it will contain those representations of amino acids that could be not utilized in the normal process. For example, X representation or the N representation of the protein. And then we also have IUPAC class. IUPAC class allows us to import those functions or utilize those functions that have the ability to define strings as ambiguous, unambiguous, and unambiguous RNA, unambiguous DNA, unambiguous protein. Basically, all of them can be utilized through these functions, but they are older functions. So we recommend that you utilize IUPAC functions, and we will obviously demonstrate that. So over here we have the previous code, but we are obviously going to delete it now. First of all, let's define our protein. Let's define our DNA. This is our DNA, and this is going to be a protein sequence. Okay, let's consider that this is a protein sequence, and I'm just going to delete this, and then we are going to create objects from it. I'm just going to put all of that in the same sequence, same variable, rather than creating a new variable all along. Okay, so first of all, we are defining a sequence object, and I would like it to be defined as a DNA alphabet, and all I need to do is pass in the DNA alphabet directly from that I imported. And I also need to define a protein sequence, and for that I'm going to utilize the original variable, it will be replaced with the new sequence object. And now we need also to provide this, and it will be protein alphabet. There we go. So now we need to just make sure that it is obviously that either it is a protein or not. So I'm going to just print my DNA. Okay, so it is my DNA, and then I'm going to also print out the alphabet. There we go. So let's just run the code, and we will see that our sequence had, so there seems to be an error. Okay, we need to save this particular file first of all, and then it will work. So let's rerun the code, and we will see our results. So there we go. Now we have MTYK is a protein alphabet, and ATGC is a DNA alphabet. So we directly defined the protein, and we directly defined the DNA. We did not do it separately as in the previous video. And we can also do the same with generic alphabet, generic DNA, generic protein, but we will discuss that in the further video that will explain the use processes of generic types. But in this video we will also demonstrate the IUPAC class, and which is very useful in my opinion. So let's go back, and I'm just going to copy this code. I'm just going to keep this code over here, so it will be useful for you. You can download this particular script from the transcription. And now I need to, I will define using IUPAC. You can download this script. Okay, so first of all, IUPAC provides various other functions such as ambiguous and unambiguous, as I said earlier. So rather than directly importing these functions, we can utilize IUPAC, and we need to just pass in IUPAC class over here, and then we need to utilize the dot operator, and then there are various functions